My name is Emad Moeendarbary. I'm a professor of cell mechanics and mechanobiology at University College London, Department of Mechanical Engineering, and currently applying engineering and physics concepts to understand the biology of the cells, how cells behave in the context of development, physiology, and diseases. Two words, neuro and degeneration. And neuro is calling neurons in your body, and degenerative means degeneration of neurons. These diseases are associated with degeneration or lack of function of neurons in your central nervous system. And central nervous system is brain and spinal cord, and the central nervous system is responsible for your cognition, sensation, and function, physical function. The underlying mechanism of neurodegenerative disease, depending on the specific disease, is varying. However, there are few knowns and there are a lot of unknowns. For example, some of the knowns are like genetic disorder and genetic mutations. The lack of diet and nutrition gonna contribute to accelerating neurodegenerative disease, but most importantly, aging is the biggest, let's say, a cause of neurodegenerative diseases. And this is very, very important for society. As we are getting healthier life and better, and we come up with better treatments for different diseases, we get older and older. The population in the world is getting older and older. The risk of getting neurodegenerative disease is getting higher and higher. So there is a correlation between neurodegenerative disease and aging. And so it's a huge risk for general population, is a huge burden for the governments to diagnose, prevent, and cure neurodegenerative disease. There are examples of neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease. There are a few others, but also multiple sclerosis can be considered as a neurodegenerative disease. And each of these diseases have been studied for years, but unfortunately, there are very small number of treatments that have been approved. Recently, there are few drugs that has been tested and passed the clinical trial for Alzheimer's disease, but it's only targeting very small number of patients. Part of the reason is like these are quite known recently diseases, but part of the other reason is brain and spinal cord or part of your body are very, very difficult to understand and examine in the human. In your central nervous system, you have these neurons that are transmitting electrical signal through this extension called axons. When you move your hands or you think, all of these actions are resultant of this electrical signal passing through these axons. So myelins is this wrapping sheets that are originated from these other cell types that neurons called oligodendrocytes. These oligodendrocytes are wrapping around these axons, which are the extension of neurons. And the first function, the most important function, is they are gonna be smoothing or fastening, accelerating the transmission of electrical signal in neurons. If there were no myelin, the electrical signal would be traveling much slower, hundreds of times to thousands of times slower. So your actions would be like very, very slow. Your cognition gonna be slow. This myelin helps transmission of electrical signal or accelerate transmission of electrical signal. This is the first obvious important function. However, structurally and biochemically, these oligodendrocytes that are wrapping around neurons are part of glial cells. Glial cells are this other type of cells in the central nervous system in brain and spinal cord that are basically gluing neurons. They are helping and supporting neurons structurally, but also biochemically. These oligodendrocytes are wrapping around neurons and neuronal extensions and helping their function as transmitting electrical signal, but also keeping them happy and healthy to function happily. The first thing that happens is like if in the context of multiple sclerosis, you're gonna lose your ability to interact fast. You're gonna lose your ability to sense. You're gonna lose your ability to respond. And ultimately you would be losing your ability to move. In the context of other neurodegenerative diseases, you would lose your ability to have a high level of cognition and memory. These neurons, as I mentioned, this myelin is connected to passage of electrical signal, and passage of electrical signals in the context of sensation, movement, but also in the context of cognition. Lack of this myelin, or let's say degeneration of this myelin or dysfunction of this myelin, gonna contribute to dysfunctioning of neurons, which means you're gonna lose ability to move, to sense, to have a high level of cognition and memory. Our model, simply we can look at these neurons, how they myelinate, and we can measure the thickness of myelin, and we can measure the quality of the myelin. Our model simply have these artificial pillars. The cells gonna myelinate them. We can see through the microscope the myelin wraps, the myelin sheets, and then we can see whether the pillars are getting myelinated, and if they get myelinated, what's the quality of myelin? Is it 1 wrap, 2 wrap, 10 wraps? How compact it is? How stable it is over time? We can directly examine this kind of features. If we can see the quality of the myelin, we can say if it is good, bad, high, medium. Then if we apply different intervention, including drugs, we can see if we can improve the score. In our assay, we have a scoring system. For example, let's say if you put hundreds of cells inside your assay, and like let's say 50%, you see 50% of them get myelinated, then if you can add a drug into our system and you can improve from 50% to 60, 70%, it means that our intervention or drug is effective. As simple as that. You just score them, look at it by microscope, and see what's the quality of myelination, can improve it with different drugs or not.